So paper for this week, something about Planaria, and then a bunch of words. That's what we're here to do today, is figure out what all those words mean. Hedgehog patched anterior-posterior polarity by regulating Wnt signal. So hedgehog and patched and Wnt are two different types of cellular pathways that cells use to communicate with each other. And that's what we're going to spend a bunch of today just going over the basics of those pathways, so you're familiar with some of the terms and the gene names that they're going to use in this paper. But first, before we get into the weeds and the nitty-gritty details, regeneration is really cool. And there are a lot of different types of regeneration that a lot of different species use. And again, unfortunately here we're going to focus on animals, but plants do lots of different types of regeneration as well. And so the next slides are my animal-focused set of slides to introduce different types of regeneration. So we have lizards, for example, that perform autotomy. That is, they break themselves, like they lose their tail when a predator attacks, and then they can regenerate their tail. It can regrow. So this is just sort of 30,000-foot view, different types of regeneration. There's paratomy. There are some species that actually reproduce by regeneration. So these are types of worms, although not at all closely related to the worms I work with, that there's actually an adult worm. There are two worms here. That's one worm, and that's the head of another worm growing sort of on the tail of the first worm, and eventually they'll break fission. They'll break off, and there will be, where there was once one worm, will be two. So that's paratomic fission, breaking. And then there's another form of paratomy that's paratomic budding, where instead of two individuals growing from the body of one individual and then breaking into two different individuals, you can also have one individual grow sort of as an outgrowth on the original individual and then break off. This is sometimes called budding. And these are examples of Planaria, so flatworms that can undergo this process of regeneration. And to get specifically at the question that the authors are going to be interested in this week, the overarching biological question is how, if you're going to do any of these forms of regeneration, if you're going to regrow a tail, if you're going to regrow another being from yourself, how do the cells in that bud know which way is up? That is, you've got brand new cells growing that are going to form a new body. How do they know how to organize? Which cells are supposed to be the head, which cells are supposed to be the tail? And you'll notice in this example that this organism is budding and its polarity is reversed. So some paratomic budding happens where anterior in the original adult is to the left, and it's the posterior end that gets the bud, but the bud itself is not in the parallel polarity. That is, it doesn't grow anterior-posterior. It grows with the reverse polarity as the parent. So the big question is, how do the cells keep track of where they are during regeneration? Or maybe put a different way, if you were a lizard and you lost your tail, how would the cells that are growing whatever it's going to be on your rear end, how do they know they're supposed to be tail and not another head? How do those cells, after the body has grown, all the somatic structures are present, it's an adult organism, and then all of a sudden it can grow another part, how do those cells know what part they're supposed to become? Different type of regeneration, architomy, which is sort of splitting down the midline. Two organisms sort of peel off from each other. So in the previous example, the anterior-posterior axis is important for the bud to know. In this case, each of the resulting planaria, these again are planaria, have to know, am I going to grow another right side or am I going to grow another left side? Because you don't necessarily want to have two left sides. Probably just as bad as having two right sides. I don't want to be biased against left-sided people. So this is the question. Really interestingly, and this is work that was done a lot in the lab of, when I was a postdoc in Maryland, the lab next to mine worked on regeneration in some worms. And they wrote this really cool review paper that looks at the different types of regeneration across a huge variety of animals. and looked at just simply summarizing. In each of these taxa, and these groups of organisms, what sorts of regeneration are they capable of? Can they do whole body regeneration, like the Planaria? You cut a little piece of a Planaria and it grows a new head, and it can be a free-living organism. Or can they just do structural regeneration, like the lizards? The tail falls off, they can grow back a new tail, but you can't cut a lizard in half and get 2 lizards. Right, so that would be structural regeneration versus whole body regeneration. Where do we fall in this list of organisms? Platyhelminth, these are worms. Nemertea, these are all worms. Annelids, mollusks can do some structural regeneration. Arthropods, insects, can do some structural, but not whole body. Nematodes, where are we? We're not echinoderms. Starfish. We're chordates. We have a nervous system. Chordates. We have a spinal cord. So this says that within the whole huge group of animals that are chordates, that there is both whole body regeneration capable in at least one of those species, and also structural. We can do structural regeneration, but not very much. We are limited to, maybe slice the tip off of a finger, the fingertip can grow back. That's about all of this structural regeneration we can do in humans.
Would be like bone bones?
Just
Would that be regeneration?
maybe a little bit.
Like when you break your bone and then they kind of...
Oh, that's healing. That's different than regeneration. Yeah, this is talking about growth of new tissue, not to repair, but to grow again. So if you lopped off a whole finger, a new finger wouldn't grow. But the fingertips, if you've been in like a bandsaw accident or something, sometimes your fingertip will grow back. And that's what we're limited to, but there's one species of chordate, I don't remember which now, thanks for asking, that actually does do but just one species that's known in chordates. All organisms that have spinal cords, they can do whole body regeneration. It's a basal. It's one of the oldest chordates too. So the really interesting thing to me and to evo-devo people is that there is a lot of regeneration capable among all of these different species. And regenerative ability is ancestral. That is, the ancestors, some of the most basal, the most distantly related to us, or the organisms that most far in the past shared a common ancestor with chordates, with humans, they all do regeneration, which means that we and all these other species lost the ability to regenerate as we evolved. Something about being human means we can only regenerate little bits of our bodies, but we can't do whole body regeneration. Wouldn't it be cool if we had restored, or if we had retained the ability to do whole body regeneration? Just tear off my arm and a new me would grow. So presumably there's a trade-off. There must be some cost to being able to regenerate yourself. There's always a cost when we lose things by natural selection. Evolution doesn't usually cause loss of traits for random reasons. Sometimes it does, but not usually. Then that's why biologists are so interested in studying regeneration in other species, because what if we learn what changes we could make to humans, what if, that would allow us to regain our ability to do some sort of regeneration, or at least maybe even reprogram cells, not necessarily living, breathing organisms, but maybe if you could take some of our cells and treat them to regenerate, like regenerate part of our liver, or regenerate an organ that we had to have removed because of cancer or something like that. What if we could regenerate bone marrow so that we didn't have to have bone marrow donations? We could have a doctor take a skin cell, make it a stem cell, and then treat it so that it would regenerate bone marrow. So those might be some of the outcomes of this line of research way in the future. So over a century ago, Thomas Hunt Morgan, do not remember the name necessarily, but this is, if you know genetics, this is Morgan of fruit fly genetics, also studied Planaria. So he's the founder of Drosophila fruit fly genetics, one of them at least, but also studied Planaria, and he had discovered that you can cut Planaria, these flatworms, a bunch of different ways and they'll regenerate. And these are some of the drawings that were published of his and later work. So the idea was, you can take the trunk, I'll show you a picture of a Planaria in just a minute. Actually, I might not I might have taken them all out. They were all parts of movies. I was going to play some movies for you, but the movie files got corrupted, so no movies. You can take a thin slice. So imagine that the head used to be up here at the top and the tail down here at the bottom. So you could take a thin slice across the body of the organism, and it will usually regrow a head in the anterior and a tail at the posterior. But he discovered that there were certain ways that you could cut maybe a thin enough slice across the body or do other things that would make, for example, that chunk regenerate. So let's say you took that part of the body and you made those two cuts throughout the top and the bottom pieces, so you just had a strip across the body. Sometimes it will just grow two heads. The head at each end, head at the anterior where it's supposed to be, a head at the posterior where it's not supposed to be. And then if you angle the cuts like this, maybe you can get the heads to grow in slightly different directions. And so he found, you just buy these days in developmental biology lab, you just buy the Planaria from Ward's Scientific or something, they get delivered to the lab, put these flatworms on a Kim wipe on a piece of ice to anesthetize them so they don't move around. And then you just use a scalpel and you make all sorts of weird incisions like halfway down the body and see if the two halves regenerate to make a two-headed, one-bodied Planaria. There's all sorts of weird things that Planaria will do depending on which exact way you cut them, and Morgan was one of the first people to study this.
Just curious, how long does it take for them to grow back?
About a week. Not too. That's what makes it nice for developmental biology lab, because you can come in 1 week and cut them, and then the next week come back and see what they've done. And so he termed all of these different types of regenerants, regenerant, the name for an organism that has regenerated from some fragment of a body. He called all of these different ones Janus heads after the god Janus, the two-faced or two-headed god. So these are all Janus heads worms. That's a term you're going to learn or hear a lot this week. I did leave a picture in. Good. So, Planaria regenerate. Here are some Planaria. This is the dorsal view looking at its back. So if you were growing this in a petri dish, this is what it would look like, this side, because you'd be looking down on it on its back. And you would see these two white eye spots, depending on which species you're looking at, it doesn't really matter. And then they kind of look cross-eyed because there are two black spots that are pigment cells that are actually photoreceptors. So they don't have eyes like we do, but they can detect light. So they can photonavigate. They know where it's lighter and darker, and they can kind of move around based on light. So it kind of makes them look cross-eyed, because the two black dots are on the interior of the two white eye spots. So that's how we know where their head is, the eye spots. And the tail is the other end. And if you looked at their stomach side, this is one of the videos I was hoping to show. Maybe I'll show it on Wednesday if I can find it again. Their mouth is actually in the middle of their body on their stomach. And there's some, you just Google it, YouTube probably. There are some really cool videos of Planaria feeding, because they actually like extrude their gut out of their pharyngeal opening, so it like comes out of the body, kind of like aliens. It comes out of the body and like grabs food particles and then sucks back into the body. And then the gonopore has to do with mating. So that's on the ventral surface. So for this paper, the most important thing to know is where the head is, where the eye spots are, because we're concerned in this paper about anterior-posterior axis. If you cut this Planaria in half, how would those cells right there know that they should start growing up and producing head-like anterior cells? And how do the cells that were immediately just right next to them in the body, as soon as that cut's made, know, hey, wait a second, I'm on the south end, the posterior end of this chunk of worm, so I need to build a new tail. Two lines of cells, they get separated by a cut. These ones somehow figure out that they need to grow that direction and make a head. And these ones that were just next to those cells need some sort of a signal that says, hey, now we're on the cut side of the head of the worm, we need to make tail. I guess I should have shown this figure earlier. So, different cuts you can make and different outcomes that they might produce. Two-tailed, two-headed, two heads, two tails, weird things. Okay. The authors will mention blastema briefly. This is a section of tissue after a cut has been made here on the anterior of a planarian. the new tissue that grows in isn't pigmented at first, and that's why it looks white compared to the original somatic tissue of the cut planarian. That's called the blastema, the new tissue that arrives to build whatever missing part of the planaria needs to be built. And so here you can see the eye spots forming about that stage. So you cut the head off of a planaria. The blastema is cells both that are thought to migrate, stem cells, thought to migrate from the body to go near the cut site, and then they divide to build new tissue. And that new tissue is the blastema. So specifically for this study, one way to think about the broad question is, why did Morgan get Janus heads phenotypes? That is, why are there some ways you could cut a planaria that they can no longer regenerate the way they should? Some cuts, they regenerate heads or regenerate tails appropriately. Sometimes, some ways you can slice up a planaria, then the cells apparently forget how to tell where they are in the body. And I already mentioned this broader goal, or more specific goal, briefly. How do the cells know where they are in the body? And if they're adjacent to a spot where an animal gets injured and regeneration needs to happen, how do those cells know what to do? So for decades, or maybe even centuries, the leading hypothesis that's true has been that, well, cells must be able to communicate with each other to tell each other what to do developmentally. So if you had a planaria with eye spots and you cut that planaria in half, one hypothesis was that there might be some sort of signal that's produced in the anterior that diffuses through the body, and that gradient would instruct cells where they are in the body relative to how much of that signal they're receiving. So in the original pre-cut organism, you have to imagine that this is the situation, that the gradient goes from high at the head to low at the posterior, and when you make that cut, then now the cells that have the lowest concentration, although it's still the same concentration as before, the cells that have the least concentration of that signal are near the cut site, so that maybe informs them that they are at the tail end of the organism and that they should produce yet more tail structure. And this is all storytelling and arm-weaving arguments. This is just what people were thinking at the time. This kind of must be somehow how it works. Otherwise, how would the tissues know what to do? So in a developmental biology class by itself, we would spend weeks talking about different signaling systems, different ways that cells send signals or communications to each other. And there are four types. We're going to focus on paracrine signaling today, the second one. Cells that are, or signals that are excreted from cells, they're sent out from the cell membrane to nearby cells. So they can diffuse through the body, go in between cells, and then tell nearby cells what to do. This is different from the endocrine and autocrine systems, which are evenly distributed throughout the body through different systems that planaria don't have. And this also differs from juxtacrine signaling, which is physical connections between two neighboring cells that don't have anything to do with signals, proteins in this case, that are secreted from a cell and can sort of diffuse in the neighborhood of cells. Paracrine or juxtacrine signals would be examples where a cell has a cell surface receptor protein and an adjacent cell has a cell surface protein that fits that receptor. So that signal, this ligand or signal, isn't free floating. It doesn't go anywhere in the body or even nearby. It can only interact with cells that are physically adjacent to it. So that would be juxtacrine signal. Paracrine basically is like juxtacrine, but you sever the connection between the ligand, the signal, and the cell surface so that it can move about. Then that little red dot could float over there and interact with a cell surface receptor that's maybe the same shape, but on a non-adjacent cell, paracrine signal. And the two pathways we're going to look at today are those paracrine type, Hedgehog and Wnt signaling. You may have heard of some of these other types. It's okay if you haven't. Notch signaling is a typical juxtacrine pathway that we would cover in a full-on semester-long just developmental biology course. But today it's Hedgehog and Wnt. Those are the two systems or pathways that they're going to talk about in this paper. Any questions at this point? So Hedgehog. You might remember, I think I mentioned it briefly, that genes are usually named after the mutations, the mutant phenotypes, the mutant traits that they have when you get rid of the gene or you mutate it. So the Hedgehog gene is important in anterior-posterior patterning to an extent in, this is Drosophila. But when you get rid of the Hedgehog protein, the embryo isn't as big, it's a little bit more ball-shaped, and it develops a lot more bristles. So the first person that looked at this said, oh, it looks kind of like a hedgehog, so they called this gene Hedgehog. And Drosophila has one, but we have paralogs. We have, that gene has been copied multiple times in, for example, humans. So we have Indian hedgehog, sonic hedgehog, desert hedgehog, and other hedgehog relatives that are paralogous genes. They're all descended from hedgehog. They're all similar to it, they just have different roles in our development. And briefly, sometimes, I think I may have mentioned this also, sometimes the way that especially Drosophila biologists want to name genes gets them in trouble. So somebody wanted to name a gene Pokemon and then Sega had something to say about it, or I don't know, something like that. Anyway. All right. So Hedgehog pathway. And I'm going to mention now that there's a lot of genes involved in these pathways. I'm going to briefly describe how these pathways work. The logic is what we're going to need to focus on for Wednesday. The logic of how these signaling pathways work. Again, getting back to this idea that we can have a set of proteins that either have inhibitory, the blunt arrow interactions with each other, or excitatory or stimulatory behavior, the arrow head. So on the left here, actually, let me describe the proteins first. So there's a gene called patched that sits in the membrane of a cell, and it is a receptor, so it's been drawn like it could fit something, and that thing that it fits is Hedgehog. So the Hedgehog gene produces a protein called Hedgehog that is one of these factors. It's a protein that can diffuse between cells. So one cell will release it, and then other cells have this receptor patched. And if the Hedgehog ligand, this protein floating in between cells, finds a cell that has a patched receptor, the Hedgehog protein binds patched. And what patched normally is doing is this protein, patched, sitting in the cell membrane, this little rectangle, normally is naturally bound to a protein that's called smoothened. And when patched and smoothened are bound, smoothened can't do its normal job. So that's why this diagram says patched inhibits smoothened. Patched is preventing, that's what that little do not enter sign or whatever it is. That in this state, patched prevents smoothened from doing its cellular job. When Hedgehog binds patched, patched changes structure. So the binding of that ligand to the receptor causes the receptor protein to change shape, which happens in proteins all the time. When they bind, they usually change shape a little bit. And that releases smoothened from patched. So it can then go do other things in the cell. Smoothened then activates the CI, cubitus interruptus protein. So in the situation where there is no Hedgehog, you don't need to know all these details really, I just want to explain it to you. When there's no Hedgehog signaling, there's no transcription. That's the end of the story, is that Hedgehog in almost all these pathways, the end goal of signaling from one cell to another is to turn on cell transcription, turn on gene expression, or turn it off. That's what cells do to each other, really, in development. So in this case, under the normal circumstance where there is no Hedgehog, this protein cubitus interruptus, CI, you don't need to know the gene name, gets cleaved in half by other proteins. And by itself, that bottom half will go into the genome and bind, what would that be called? There's a gene here, just to the left of the gene is, yeah, it's the promoter. So this cubitus interruptus gene, in its cleaved form, is a transcription factor, but it's the top half of this protein, the part that gets cut off, is important for it to turn on gene expression. So without that part, this bottom half goes into the chromosome, into the nucleus rather, binds to its binding site, but can't turn on gene expression. It can't activate gene expression because that's the bit that this top half does. So it just binds to the DNA and prevents transcription from occurring. When Hedgehog binds, then cubitus interruptus doesn't get cleaved, doesn't get cut in half. So the whole protein goes into the nucleus where it turns on transcription. And that's the logic behind the way they've drawn the pathway as it's been drawn. And we're going to need to use this sort of logic to understand Wednesday's paper, which is why I'm spending so much time describing what is going on inside the cell. So hopefully between now and Wednesday, if you're confused or unsure, you can try to tell yourself that story between the cartoon and the way that the pathway is drawn.
What's the CBP in the writing?
I don't remember what CBP is. I think it's a cubitus interruptus, I'm going to guess, cubitus interruptus binding protein. It's probably a transcriptional activator that binds to that top half of cubitus interruptus that's missing when Hedgehog isn't bound. And again, that's usually how these transcription factors work, is that the transcription factor itself binds to DNA, but it has to associate with other proteins that associate with RNA polymerase to bring it to the gene to start transcribing and producing protein. Transcription and then translation, producing protein. Whew. Okay, so that's Hedgehog signaling. Cell receives a signal, signal causes some sort of transcription to occur. This says transcription of Hedgehog response genes. Makes sense to me. When Hedgehog is there, Hedgehog response genes turn on. And on Wednesday, find out what some of those Hedgehog response genes might be related to anterior-posterior development. The other protein that they're going to talk about, which I'm not going to spend as much time talking about the cartoon, it's a similar process, is Wnt signaling. Similar story. Ligand, Wnt, a small freely diffusing protein out in the intercellular milieu, can bind to a different receptor, frizzled, and that binding causes another cascade of cellular events that causes, importantly, the protein beta-catenin, which is also a transcription factor, to go bind its binding sites in the nucleus and turn on gene expression. So the story is essentially the same, it's just different proteins. So we're going to look at Hedgehog signaling, we're going to see Wnt signaling. All right. There are a few terms and techniques that they're going to use, so I want to go through those briefly. Ish. That reminds me, I do have a video to show. So I better get prepared for that. So one thing that these scientists are going to do is to look at what they call an EST library, which stands for expressed sequence tags. Really what this means is messenger RNA. So an expressed sequence tag is a small chunk of a gene. And the way that they're prepared is that you extract messenger RNA, which I'm going to describe as having lots of adenosines on its 3' end because messenger RNAs have lots of A's on their 3' ends, one end. Those are converted by a process called reverse transcription, which is the reverse of transcription. So it's taking RNA molecule and making it DNA, which is not the way the central dogma works. The central dogma says DNA gets transcribed and you produce an RNA molecule that encodes a protein. Here we're going backwards from RNA to DNA, and it uses this enzyme reverse transcriptase. Reverse transcriptase is not an enzyme that we have, because we don't need it, because we don't do RNA makes DNA. What organisms do RNA makes DNA?
Why don't viruses do it? Are the organisms?
Well, okay, so we could debate whether or not viruses are alive or whether they're organisms, but viruses sometimes have RNA genomes, but they make copies of themselves to insert into the host DNA, so they have to be able to turn their RNA genome into DNA to put it in the host chromosome, to insert it into the host chromosome. So viruses produce reverse transcriptase, and so that's how in vitro, that is in the laboratory in a tube, we can isolate reverse transcriptase, the enzyme. We can combine it together with messenger RNA that we collect from organisms and make viral reverse transcriptase, turn the RNA back into DNA. There are subtle reasons why it's important that this be DNA and not RNA for a study, but that process of reverse transcription, you can turn an RNA molecule back into DNA. This is called cDNA for complementary DNA, but for all intents and purposes, it's a DNA molecule that was encoded by an RNA molecule. And this cDNA can be referred to as an expressed sequence tag. It's just another word. What it means is if you have an EST, if you have a specific DNA sequence that was messenger RNA and now you've converted back into DNA, cDNA, you know that at some point that sequence was messenger RNA. You know that this sequence that you collected from an organism was from a gene that had been transcribed. That's the point of ESTs and cDNAs, is that they tell scientists that collect them, this is a sequence of a gene that had been transcribed in the organism I was studying. Because there's lots of DNA that is not protein coding, and scientists, at least in this study, don't care about non-protein coding DNA. They want to know about genes that are involved in anterior-posterior patterning. So what they said they did was that they screened, this is their words if I remember correctly, they screened an EST library. And I think they didn't provide many references to this, and they didn't provide data from this, so this is my best guess, educated guess, is that you collect let's say that a regenerating anterior is in red. There's some eye spots. Ooh, red doesn't show up very well. Anterior, the head is red in this. Maybe I just can't see it because I'm kind of colorblind. But and then you take, let's say you took a posterior regenerant, and you collected cDNA, that is messenger RNAs that have been turned back into DNA, also known as expressed sequence tags, from both of those animals. And then let's say you determined the DNA sequences of all of those cDNAs, ESTs, messenger RNAs. The question that they're asking, if they do this, which again is what I think they did, is, well, what sort of genes are being turned on in this organism? Can we think of some classes of genes that you would expect to be present? You just take that whole organism, the whole Planaria, grind it up, get all of the messenger RNA out, and sequence it. That's basically what they do. They want to know what genes are being turned on right now in this whole bunch of tissue. So what sort of genes are we going to find? Okay, so developmental genes. What other genes are going to be turned on? I mean, our cells are always doing transcription. They're always making proteins. So we're going to get transcription factors. We're going to get the genes that encode microtubules. We're going to get DNA polymerase. We're going to get enzymes that do normal cellular functions. We're going to get genes that are involved in energy production in cells, right? There's going to be a ton of genes being expressed in the whole body of a planarian. There are lots of genes, because that's hundreds or thousands of cells. All of those cells are producing a bunch of different types of enzymes, proteins. So how does sequencing the anterior regenerant by itself and the posterior regenerant tell us anything about what genes are important in development? So let's say you sequence this whole anterior side and you see that there are 9,368 different genes being expressed. And in the posterior side, let's say you sequence that cDNA library and you see 7,382 genes, different genes, in the whole body of the animal at that point in time. So you've got a list of genes from organism A on the left and a list of genes from organism B on the right.
If they look at the list of genes and they find some that are only expressed like in the anterior or only in the posterior, they would know that they're responsible or they're at least like playing an important role in the development of that part of the body.
Right. So the trick here is they're looking for differences in gene expression between the left side and the right side. So if there are genes that are specific to anterior development, they should only be part of those 9,368 genes. And in the planarian on the right, if there are genes that are important in posterior development or patterning, then those should be a subset of the 7,382 genes. So if you draw a Venn diagram, most of the genes that are expressed in both of those planaria should be the same. They're the normal, everyday RNA polymerase, DNA polymerase, actin, myosin, well, maybe not myosin. Well, probably myosin. In other words, they might found that there are 7,000 genes that are shared in common between those two EST libraries. It's the differences they're looking for, as Pari is suggesting. The genes that are expressed in an anterior regenerant that are not expressed in a posterior regenerant are likely the genes that are involved in anterior patterning, and likewise the genes that are unique to this set of transcripts, messenger RNAs, that aren't present over here tell us something about what genes identify genes that are probably involved only in posterior development. And that's how they discovered or came across the idea to start working on these signaling pathways, because one of the genes they found in that sort of an analysis was Hedgehog. Or was it Wind? One or the other. So that's how they got into the idea of studying signaling pathways. Was they did that sort of an analysis and they found that in one library, one of these signaling pathways was much more heavily being turned on than in the other one.
How expensive is to do this, or how many times do they do it to make sure they get like the same or similar number of genes every time?
That's a good question. I don't know, because EST screening is an older approach. There are newer ways to do it nowadays with deep sequencing. So I don't know how they replicated that. You probably do it 3 or 5 times and see if you see a similar, on like 3 or 5 times on an anterior regenerant and 3 or 5 times on a posterior, and then really do an intersection of what common genes are turned on and turned off.
And then how do they deal with, I mean, I don't understand all this exactly, but
Yeah.
if they have, if they're going from RNA to DNA, how do they know whether the RNA wasn't put by 2 introns, or how do they work that? If the ending RNA was put together by 2 introns, how do they make those?
Right. So they would still, so they don't need to make, so cDNA does not contain introns, so they're not remaking the original gene when they make a cDNA library. The only thing that they know is that whatever is in a cDNA was an exon. There was a part of the DNA that was expressed. It was turned into a transcript. But yes, they don't need to go back to making an intron-containing version of the cDNA. The other thing that is important, oh, look, there's an RNA right here, is the other way you start making a cDNA library is you only extract from a cell genes or messenger RNAs that have the poly-A tail, because those are messenger RNAs. They don't get any other sorts of RNAs that are in the cell, like transfer RNAs, ribosomal RNAs. So they, by doing that selection just for RNAs that have that poly-A, they know that they're looking at genes that have been transcribed, that are protein coding potentially. In the past week or maybe two, we've talked about this RNAi approach. It's a way to get rid of gene expression. So there are two different ways to turn off a gene. One is to knock it out, that is to remove the gene itself from DNA, from the chromosome, just physically cut it out and remove it. But there's a cheaper and easier way that's not necessarily as efficient, which is to use RNAi. And so people have mentioned this. I wanted to give a little bit more detail into how RNAi works. And what RNAi is it's a method that cells use to destroy invading DNA, essentially. And the way it works is, and this is a cellular response that our cells evolved to be able to defend ourselves from invading nucleic acid sources, invading chromosomes from viruses and bacteria, for example. So what you do in the laboratory is we can design these microRNA molecules, that form a particular secondary structure. That is, they base pair with themselves, so they hydrogen bond with each other in a certain shape. And that shape is called a hairpin, because it looks kind of like a bobby pin or a hairpin. And messenger RNA, or microRNAs, RNA molecules that have that shape are bound by a protein called dicer. And dicer is a protein that binds these RNAs, and dicer is an enzyme that cuts that single-stranded but base paired to itself RNA molecule into two different single-stranded molecules. Basically, it clips off the hairpin loop there, so that it becomes a double-stranded RNA molecule. And that strand, one of the two strands, then gets bound by a different protein complex called the RISC, R-I-S-C, for RNA-induced silencing complex. And what that RISC complex does is it uses that messenger RNA, or that microRNA molecule, they hate it when they both start with M, microRNA molecule, short, usually 18 to 20 base pairs long. That protein uses that sort of as a guide to look for other RNA molecules in the cell that are complementary to that sequence. That is, that would base pair with this single-stranded RNA molecule that's bound to this protein. So this protein basically goes around the cytoplasm of the cell and tries to find messenger RNA molecules that match its template, this single-stranded RNA that's bound to it. And when it does find a match, when a matching RNA base pairs with this microRNA, then that RISC complex, RNA-induced silencing complex, basically targets that long messenger RNA molecule for destruction. It basically cuts it up into little pieces. So that's the long version of the short story. Short story is, you can design an RNA molecule and order it from a company, and they will ship it to you. You can design an RNA molecule, a gene, that when it gets transcribed, makes a single-stranded RNA molecule, like most transcripts, that will fold on itself to make the structure that the dicer enzyme recognizes and will bind to and cut. And you design this gene also so that your microRNA gene has a stretch of sequence that's identical, complementary to the gene you want to turn off. So you design this gene for two purposes. One is it has to be identical or complementary in sequence to a gene you're trying to turn off. And it then also has to be able to fold up to this particular shape. And if it can make that shape, you'll be bound by dicer and processed so that your single-stranded RNA molecule that will target the gene you want to turn off binds the RISC what's the word I'm trying to think of? Lots of proteins. And complex, C for complex, risk complex, so it's kind of redundant. That gene then that you've designed will target and destroy every messenger RNA, or almost every messenger RNA in the cell that is complementary to that sequence. So design a gene to turn off, to destroy the transcripts from a gene. The reason that RNAi then isn't perfect, well, I don't know, why isn't RNAi perfect? Why doesn't it work perfectly effectively at totally eliminating production of proteins from your target gene?
I would say it's a matter of like that collision happening and all that stuff.
Yeah, so it's,
Finding-
but yeah, it's kinetics, partly. How many messenger RNA molecules have you gotten this organism to transcribe? Are there enough of them to target enough of those transcripts to destroy them all? No biological process is perfect, so even with efficient RNAi, you're not necessarily going to get rid of every last molecule of transcripts. So there's usually some baseline small level of transcription from genes that you've targeted, just because this is not a perfect world.
Maybe because RNA is also not super stable and it
Right, so yeah, there are other issues with RNA too.
doesn't have to be fast enough.
Yep. And if the cell's really churning out a lot of transcript, that's another one of those kinetics things. Do you have enough RNAi to knock down all of the transcripts? This is why knockouts are better. If you get rid of the gene that encodes this RNA, you never make any of it in the first place. So microRNA or RNAi is a good solution. It's easy to apply. It's much technologically easier for some organisms to get RNAi to work than it is to genetically engineer the genome of the organism to remove whatever gene it is you're trying to get rid of. It's especially useful in developmental biology.
Wait, what was the difference between siRNA and RNAi?
There are many different short RNA-based gene knockdown mechanisms that are used by different organisms. So some organisms use, and actually that's suggested here down at the bottom. So sometimes targeting for cleavage, other times the fact that you have this RISC complex bound to the messenger RNA doesn't destroy it, it just keeps it from being translated because there's this huge protein complex on the RNA that doesn't let the ribosome finish translating the protein. Plants do a version of RNAi that's not the same as this RNAi mechanism. So there are lots of different tricks that organisms have evolved over time to try to prevent invading RNA basically from getting into their cells and their genomes. So microorganisms are the ones that evolved the RNAi process, and presumably that was to knock down the expression of, say, if that was a viral transcript or a transcript of another bacterium. So we find all sorts of really cool things in other organisms that we co-opt for our own use, like reverse transcriptase and RNAi.
So they decided to go with this instead of the knockout.
Right.
Because the knockout would they would be fatal to the Planaria.
In theory. So that's where I was about to go, and that's exactly right. Most of the time for developmental genes, they're essential genes. If you made a knockout, they wouldn't develop in the first place, so you wouldn't have an organism to study. So if you let an adult organism grow and develop, then you can provide RNA to it and see what happens. So for example, if you cut a Planaria in half, and then you provided these short RNA molecules to try to prevent some genes from turning on, you could see how does that knockdown of those genes at that point in time, early regeneration, let's say. You could find out which genes, when we knock them down, prevent regeneration, or which genes, when we knock them down, make worms, Planaria, regenerate a tail instead of a head, or a head instead of a tail. And that's how you can figure out what genes, the genes that you're trying to knock down, the target genes, you can find out if they're involved in this process or not. Will you talk about qRT-PCR for us on Wednesday?
Yeah.
I know what it is. I've never done it, so I can talk about it. But it's an approach they use a lot. So let me just say one thing about it. If you want to go into detail on Wednesday, you can. It's just a method of measuring gene expression, quantitating gene expression. It also uses that RT up there in the qRT-PCR. The Q stands for quantitative. It means you accurately, quantitatively measure an amount of transcription. And the RT we've already seen once before today. Reverse transcriptase. So it takes messenger RNAs and you use reverse transcriptase to make DNA out of it, and then you can measure gene expression. So this is basically the new version of EST library analysis. Instead of pulling all the messenger RNAs out from an organism and figuring out which ones are more abundant and which ones are less abundant, you can measure gene expression more specifically of an individual gene at a time. So if you wanted to compare pre-amputation, post-amputation planaria, if you've got one specific gene, you want to know how does the amount of transcription change from situation before amputation to situation after amputation, you can target that one specific gene and find out if it's changing in its expression, for example, when anterior regeneration starts or when posterior regeneration starts. The last concept is epistasis. This is a tricky one to define, so I'm going to work through it in two ways. I've always found this a really hard concept to teach, is what I'm saying. So I'm going to try to define it two ways, and we'll see what happens. This is, if you want to tune out and you haven't already, this is a good time to tune out because this is a very minor part of the manuscript. And they don't directly call it epistasis. So if you pay attention and you figure out where this is all relevant in the paper, I'll be super impressed on Wednesday. So please try. Here's the idea. What would happen? One moment, please. We saw this pathway last week, right? Fly, pigmentation, something. So that's the reason I've chosen this pathway. The concept of looking for epistasis is to try to figure out what order genes work in a pathway. So for example, in this pathway, we know that tyrosine hydroxylase produces an intermediate molecule that can be converted by the yellow enzyme to produce melanin. And we know that DDC turns dopa to dopamine, which is another precursor used by some unknown enzyme to produce a brown pigment, or dopamine can be converted by the ebony enzyme to produce another pigment that produces yellow pigmentation. So that pathway has already been worked out. But the question of epistasis is, people that are asking epistasis questions don't know the order of enzymes in a pathway. That's what they're trying to figure out. Which enzyme works first, then which enzyme works second, then which enzyme works third, in this case. So for example, here's one way that an epistasis experiment would work. So you knock out tyrosine hydroxylase, or knock it down, RNAi, TH. What happens to pigmentation in that case? If you get rid of that first enzyme? Zero tyrosine hydroxylase equals, what sort of pigments get produced by the fly? Yeah. Because that's the first, we know now that that's the first step in the pathway. So if you get rid of that enzyme, none of these other pigments can get synthesized. Yellow has nothing to work on because there's no dopa produced. Dopamine or dopa decarboxylase doesn't produce dopamine, so you don't make brown pigment because there was no dopa produced, because tyrosine hydroxylase is absent. You make no yellow pigment because there was no precursor. So that's one. So these are all, the epistasis experiments are all mutagenesis experiments. You get rid of a gene, knock it down or knock it out, see what the effect is. And then ultimately the point of the epistasis experiment is to compare the pattern of the outcomes of the different experiments. So we've got 2 more experiments to work through, and then we look at the pattern. So let's say instead we knocked out, in a different color this time, ebony. So if we get rid of ebony, what colors might this Drosophila fruit fly include?
Brown.
Right, it can still, so tyrosine hydroxylase is here. This is just a single mutation. So you can still make black, you can still make brown. This pathway is blocked at the step towards making specifically yellow. So you get black and brown when you knock out ebony. So last question then is, what if you knock out both tyrosine hydroxylase and ebony, double mutation? So you do each of the single mutations, and you do a double mutation, double knockdown. Get rid of both at once, both TH and ebony. Then the result is no pigment. This is an epistasis experiment. It tells us which of the 2 enzymes works first. Which one works first? Well, the important thing to remember is that we don't know the order of the enzymes at first. So the information we're looking at is simply that. If you don't have tyrosine hydroxylase, you don't make pigment. If you don't have ebony, you do make pigment. If you combine those 2 mutations, you don't make pigment. So the analysis is you look for the combination of outcomes that are the same, and the gene that's common to both of those is the one that's upstream. That is, if it's the first, or if it's an earlier gene in the pathway, if its effect by itself, will be the same as the effect of it plus any downstream mutations. Because if you get rid of this, you could do anything else. You could knock out all of those genes. That wouldn't change the phenotype, the outcome, because you've gotten rid of the upstream-most protein. So this is how you find out if 2 different genes are acting in the same pathway. If you knock out 1 and you see 1 phenotype, and you knock out 2 and you see the same phenotype, that means that the common one is upstream and the other mutation must be downstream in the pathway. So that's an epistasis experiment. Normally done by making 2 single mutations and then combining the 2 mutations at the same time and figuring out if, for example, if that result had been black and brown pigment made, then that would mean that the ebony protein was upstream for contrast. So it's one of those experiments that makes a lot of sense after you actually know what the answer is. But it's hard because this is what geneticists do in the first place to figure out the order of genes. And I was going to show you this video that I spent a couple hours filming yesterday, which was kind of ridiculous, but now I think I've flogged that dead horse enough. But you can watch it if you want to because the URL's there. It's a silly analogy that I built out of dominoes to represent the different steps in a pathway and removing different dominoes and seeing what the effect was on. So if you like that, I'm going to use it for my Biology 102 class, my genetics class. So it was worth my doing, but if you think you want another view of what epistasis means or how the experiments are done, you can check out that short video. Lastly, I put together, the last time I taught this paper, I put together this worksheet that may help. So it's already on Canvas right now. If you want to download it and fill in the blanks as you work through the paper, it might help you understand the logic behind the experiments. Might. So what this is it's got the Hedgehog pathway members that are relevant to this paper, and it's got the Wnt signaling beta-catenin pathway here. And you can't read it but I know what it says. It says RNAi phenotype, wild type role, and something that I actually don't remember. So in other words, what they're going to do in this paper is they're going to RNAi Hedgehog, patched CI, cubitus interruptus, and beta-catenin. So if you write down what they saw, what the result was, what the phenotype outcome was when they did each of those individual experiments, then that should tell you what the wild type role is. Remember, if you knock down a gene and you see one effect, that means its wild type role of that gene that you knock down is the opposite effect. This may be a useful structure for understanding exactly the logic behind all of the experiments they do. Maybe. I found it useful, but I'm just one person.